Welcome to this webinar titled Current Bacterial Endotoxins Test and its Intended Use, hosted by Biopharma Asia magazine and presented by Karen Zink-McCullough, owner, principal consultant at MMI Associates. Hi there, Karen. Thanks for joining us.
Hi, Marcus, and thank you for having me. This is, I think this is going to be a good webinar. I'm so excited to hear Veronica's presentation. I'm excited about this. Thank you for having me.
Great, thanks a lot. And this presentation, of course, will be followed by an industry perspective and presented by Veronica Wills, Manager, Technical Services at Associates of Cape Cod. Hi there, Veronica. Thank you for joining us as well.
Hello, thank you.
My name is Marcus. Oh, sorry, yep. My name is Marcus, and I will be the moderator. So before we begin, I would just like to inform our viewers that there will be a live question and answer session after both presentations near the end of the webinar. So audience members, you can send your questions in via the Questions tab at any time during the webinar, and that's located directly below your webinar screen. Please also state in your questions which presenters your questions are for. Now with that, please allow me to introduce our first presenter. Karen Zink-McCullough is the Principal Consultant at MMI Associates, a firm focusing on GMP and microbiology in the drugs, biologics, cell therapy, and medical device industries, with a specialty in the Bacterial Endotoxins Test, BET. Ms. McCullough was the first to publish on the use of BET as a replacement for the rabbit pyrogen test for small volume parentals. She has published over 50 articles and book chapters and is the editor of the book, The Bacterial Endotoxins Test, a practical guide. Karen is well respected as an international speaker on GMP and the BET. Ms. McCullough is the chairperson of the LAL Users Group, is a United States representative to Working Group 2, TC209 of the revisions of ISO 14698, and is a member of the United States Pharmacopoeia Expert Committee on Microbial General Chapters. Karen holds a bachelor's degree in the bacteriology from Rutgers University and a master's degree in molecular biology from the University of Oregon. And with that, I'll now hand over to our first presenter. Welcome, Karen.
Thank you, Marcus, and welcome to everybody who's on the line. Appreciate the opportunity to do this. Veronica, you and I have done this a bunch of times, haven't we? Okay, so I'd like to start out and talk about what I'm trying to convey here, and it's about the intended uses and common misunderstandings. You know, sometimes we have a test and we try to attribute things to the test that it really can't do, and we sometimes ignore things that the test really can do. So what I'd like to do is talk a little bit about this and see if we can come to some sort of put it into some sort of context. So the first disclaimer is that the views in this presentation are only mine, and they do not reflect the opinions of any organizations with which I may be affiliated. So having said that, the goal of this is to provide some context for BET. Pretty simple. So all pyrogens are endotoxins. That's one of the things that people would like to say. Well, the answer to that is false. Okay, there's all kinds of things, including viruses and chemicals and all kinds of stuff that can elicit a fever in mammals. Endotoxins from gram-negative bacteria really aren't pyrogens in a direct sense. What they do is they initiate the pyrogenic response, the innate immunity response in mammals. But they are initiators, and so we tend to call them pyrogens, but they themselves really aren't. But endotoxins can be really potent pyrogens, and they are the pyrogens that are most often identified in pharmaceutical manufacturing environments. And so that's why they become a focus, and that's where the Bacterial Endotoxins Test has its utility. What about all endotoxins are pyrogens? Well, that's not true either, because the degree of pyrogenicity or the potency of a gram-negative bacterium has a lot to do with its pyrogenicity, and that has to do with the fine chemistry of the lipid A portion of the lipopolysaccharide molecule. So, for example, there's an organism called Heliobacter pylori, which is associated with ulcers. Well, it's about 1,000 times less pyrogenic than E. coli endotoxins, which is the current standard. And the LPS from the gram-negative bacterium Pseudomonas aeruginosa, which is more likely to be found in a pharmaceutical environment, is about 15 times less potent than the LPS derived from E. coli. And so if you tend to look at these things from a structural standpoint, we find that they differ. There's a certain common backbone structure, but they tend to differ in the number of acyl or fatty acid groups. And the E. coli, that is our standards, perhaps the most potent. It has 6 acyl groups. But what we'll see on the next slide is that there is a curve here. So if you have 6 acyl groups, you're generally pretty potent in terms of being a pyrogen, as indicated by IL-1 release, which is the sort of internal pyrogen in mammals. And as that acyl chain changes and it becomes fewer acyl chains or even more acyl chains, that potency decreases. So not all endotoxins are pyrogenic, at least to the same degree. Next one, all gram-negative bacteria have endotoxins. Well, that's not true either. There is a class of gram-negative bacteria called Sphingomonas, and there are other gram-negative bacteria that do not have lipopolysaccharide in their cell membranes, and so they don't have the traditional LPS endotoxin. Sphingomonas have sphingolipids in their cell membranes and not lipopolysaccharides. And so therefore, that's a class of gram-negative bacteria that does not have endotoxin, certainly as it's described in terms of lipopolysaccharide. And in fact, if you take a look at these data, and these data were kindly provided by Nicola Reed and presented at a conference back in 2019, if you look at that pie chart on the left-hand side, what Nicola Reed and her colleagues at Charles River did is they looked at all of the organisms that they isolated from water system. They did a large study, and they speciated them. And if you look at this, it's a little bit hard to see, but I will tell you there are no enterics in this bunch, okay? And about 25% of them were Sphingomonas. Sphingomonas don't have LPS. And then you can see on the table over here where the rest of the distribution is. So in this case, what we have here is a water system where about a quarter of all of the isolates that were identified in this study were Sphingomonas that do not have lipopolysaccharides. So we recognize that not all gram-negative bacteria have LPS, and the Charles River data then would suggest that 100% of the detective endotoxins activity comes from about 75% of the recovered and viable organisms. And so the other thing is we have to recognize is that it may not be that all organisms that have endotoxins are recoverable. So there could be something in there that's presenting us with endotoxins that aren't growing out. We also have to remember that endotoxins remain active after the bacteria are dead, and that the gram-negative bacteria generate these things called outer membrane vesicles, and we'll talk about that in a second. Those outer membrane vesicles are not viable organisms, but they contain endotoxins. And so it's really, really hard to correlate BET results with bioburden results. People often ask, well, how much endotoxin is in the average gram-negative bacterium? And the answer really is it depends. It depends on the bacterium. It depends on the growth conditions. It depends on an awful lot of stuff. And so it's really difficult to assign an average contribution in terms of activity from gram-negative bacteria. Anyone, because they're all a little bit different. So let's talk about something called autochthonous endotoxins. Okay, that's a little bit different. A new word that took me forever to learn how to spell this word. But really, this is what the LAL test is all about. This is what the LAL test's intended use is to look for autochthonous endotoxins. Autochthonous meaning indigenous. So autochthonous endotoxins come from organisms that have adapted to their environments. And these organisms have adapted to pharmaceutical environments, and largely these environments are oligotrophic, meaning that they are low nutrient environments. Now let's think about this for a second. The RSE, CSE, LPS that we use as a standard is grown in a medium, it's grown in a laboratory, in a certain well-defined medium. It's extracted, it's purified under very well-defined conditions. And that's very, very different than the average autochthonous endotoxin that we find in a pharmaceutical manufacturing. Autochthonous endotoxins is kind of a weird term. It really is endotoxins from autochthonous organisms. Okay? When you look at the organisms found in a pharmaceutical environment, and as we saw from the slide before, a good number of them, at least the ones that have measurable endotoxins, are from the genus formerly known as Pseudomonads. And that reminds me of Prince, the artist formerly known as Prince. So if we look at endotoxins and we look at endotoxins in the real world, they don't exist as monomers, just they, like the standard does. They exist really as part of the outer leaflet of the outer cell membrane of gram-negative bacteria. And they are embedded in the outer cell membrane. They are not free-floating. And so if you look at this, it's on the right-hand side is sort of a graphic about what the average LPS looks like. And if you look at this, okay, you have on the right-hand side the lipid A. Now, lipid A is highly anionic. And what happens with this is that it buries itself in the cell membrane. So you can look over on the left-hand side, and you can see that the lipid A, which is the little black parts in there, the little black parts, that's embedded in the membrane. The O antigen, or the O polysaccharide chain that's sometimes used to type bacteria, that's the part that sticks out into the environment. So when we see endotoxins in the real world, when we see endotoxins from autochthonous organisms, what we generally see are either these outer membrane vesicles, or we see intact bacteria, or we see cell wall fragments from bacteria that may have been killed. But rarely do we see free-floating purified bacteria. LPS. So let's look at LPS and endotoxin outer membrane vesicles. And if you look down here on the right-hand side of the slide, you can see that there's an organism and there's a little bud coming out of it. It almost looks like a yeast bud. And what happens is that that bud eventually pinches off, and that becomes the outer membrane vesicle. Now, outer membrane vesicles actually serve as a big portion of the cell's life cycle. It's a way to transport things out of the cell. And these little guys here, these little guys are free floating. But what they are is they are the outer cell membrane. So the outer cell membrane buds out, and it encapsulates a little bit of the periplasm, and it floats off. And these little outer membrane vesicles can start biofilms. They can be the anchors for biofilms, but they can't reproduce. They don't have any mechanisms so that they can reproduce. If you are to look at them in the microscope, you would see that you can see them. They're little tiny spheres, okay, and these are the outer membrane vesicles. The graphic in the middle shows how that budding happens. But if you look at the picture on the left-hand side, that's a picture of purified lipopolysaccharide. And purified lipopolysaccharide forms micelles. Those monomers form micelles and ribbons because, remember, there's a hydrophobic end, the lipid A end, and a hydrophilic end, which is the polysaccharide end. And so these purified lipopolysaccharide molecules, in order to survive, essentially, naturally form these micelles, okay, based on the hydrophobicity and the hydrophobic and hydrophilic nature of the LPS. And so if you look at these two things, they're very, very different. And it's the stuff on the right-hand side that we normally see in our products, not the stuff on the left-hand side. So the stuff on the right-hand side, those are endotoxins from autochthonous organisms. So what happens when an organism finds its way into an environment that it doesn't like, an environment that's stressful, an environment that if the organism doesn't do something, the environment will kill it? Well, gram-negative bacteria, as well as pretty much all bacteria, have a way in order to modify their lipopolysaccharide in response to the environmental stresses. So, for example, we know that lipopolysaccharide is highly anionic. It has a net negative charge. And the lipid A embedded in the cell membrane, okay, what happens is it needs magnesium or another divalent cation in order to sort of neutralize that net negative charge. So if that bacterium is growing in an environment that provides those divalent cations, then the cell membrane is likely to stay intact. And the question is, what happens if those divalent cations are removed? If those divalent cations are removed, then those highly anionic lipopolysaccharide molecules will likely, cause the cell membrane to fall apart, lose its integrity. So unfortunately, I neglected to put a reference on here, but this is a reference from Bonington and Kuhn, and anybody who wants that reference, I'm more than happy to give it to you. And what this does is it looks for bugs in the real world. It says, how do these organisms adapt? And these organisms adapt through something called a two-component system. And what happens is when the stress level in the environment gets to the point where the organism is likely not to survive, the two-component, it's called a two-component signal transduction system, but these two-component systems, they upregulate the transcription of certain genes associated with lipopolysaccharide generation. And what they do is they generate a different lipopolysaccharide. In this case, on the left-hand side, this is the lipopolysaccharide that has to be stabilized by magnesium. On the right-hand side, you can see that there's all kinds of substitutions here. I mean, it is 4-amino, 4-deoxy-L-arabinose, and zwittered ionic phosphoethanolamine. Oof, I had to read that off of the paper here. But what those do is it replaces the so-called salt bridges on the wild-type organism with sugar bridges. And these sugar bridges allow the cell membrane to stabilize and therefore allow the organism to survive. So these autochthonous organisms can have lipopolysaccharide structures that are very different than the wild-type structures, which can be very different than the E. coli that we tend to use in order to make a standard. Another false statement, the BET LPS standard is representative of organisms autochthonous to pharmaceutical operations. So back in the day, if you read, there's a great series of books that were generated as the proceedings from the Woods Hole symposia that happened back in the late 70s and early 80s. And E. coli was chosen back in the day as a model strain for the calibration standard because it was determined to be typical of gram-negative organisms. And why did we need a calibration standard? Well, back in the day, okay, when people were making different kinds of lysate, we needed a way to standardize and calibrate the different lysates. Because even though LAL is essentially, it's the same thing, only different, right? They all come from the horseshoe crab amoebocytes, but the formulations are different and this and that and the other thing. All that's proprietary, but there needed to be a way to calibrate all of this. And that's where that standard arose. Now, at the time, the standard was decided upon by a large group of people. There were clinicians, and there were government people, and there were industry people, and there were academics. And this group decided that E. coli may have been typical, but even back then, people like us in the pharmaceutical manufacturing business saw that perhaps this was not the right choice for us, that these bugs don't normally show up in our manufacturing. They can, but they don't normally show up in our manufacturing, and therefore perhaps are not the best representative of all of the universe of organisms that might find its way into pharmaceutical manufacturing. So we knew, as did Florence Seibert from stuff she published in 1923, that it really was non-fermenters. It was the organisms then and formerly known as pseudomonids that were most likely to be found in the water systems. And those are the ones that really we are worried about. Those were the ones that we were worried about back in the day, back in 1923, and those are the ones that we're still worried about today. We need to remember that the LPS from the standard, it's not OMVs from the standard bug, it's not cell wall fragments from the standard bug. What it is it's purified lipopolysaccharide, purified and formulated lipopolysaccharide. It's formulated for stability, which is unique for a standard. It needs to have all of this stuff in it, but it is not at all representative of what we might find in our products. Another one, recombinant reagents really are the same as LEL reagents. Well, that's not true either. Okay, we may have a cascade, and I should have put a picture in here, but I didn't. But everybody on the phone probably can visualize this cascade of factor C, and factor C gets activated by the lipopolysaccharide, and then the active factor C in turn activates factor B, which in turn activates the proclotting enzyme, which in turn leads us to, depending on your test, an increase in color or an increase in turbidity. Well, in real life, these amoebocytes from the limulus horseshoe crab, and you can see these pictures on the side, they have large and small granules in them that are lysed, and the contents of those granules are released into the lysate itself. Now, different lysate manufacturers, Veronica's company and others, have different ways of taking this gamish of all of these things that are in the lysates and formulating them into a lysate that we can use day to day. But essentially, the source of those lysates is these granules. And there's a great paper that was published in 2002 by Dr. Ironagua, and he identified at the time at least 43 other substances. And this is, 18 years ago. He identified at least 43 other substances, and that was excluding the clotting stuff that we needed, the clotting factors, in these lysates, in these amoebocytes. And all of this gamish included protease inhibitors and lectins and antimicrobials and all kinds of other stuff. And the question then becomes, recombinant factor C doesn't have all of that kind of stuff. So while recombinant factor C may have the factor C part of this cascade, and the recombinant cascade reagents might have all of the component parts, they're still missing a lot of what is in the background of these amoebocytes. And I guess I did do a picture. So here's the normal cascade. You have the factor C making an activated factor C, which then activates factor B, which activates the clotting enzyme, and depending on the kind of test you have, does one thing or another. But look at this over here. This is another pathway called glucan. So let's think about this for a second. What we're looking at when we look at amoebocytes is we are looking at the innate immunity system of the horseshoe crab. And the horseshoe crab is not only susceptible to gram-negative bacteria. The horseshoe crab in nature is susceptible to a lot of stuff. In this case, we also know that it's susceptible to glucans, and that glucan here can kick off this pathway. It doesn't start up here, it starts in the middle, but it can activate the pro-clotting enzyme and turn it into a clotting enzyme. And what we can get is a reaction here that is not generated by endotoxins, but in fact is generated by these glucans. The good news about the recombinant reagents, and up here we have the recombinant factor C reagent, and as you can see, it only has factor C, and the activated factor C then acts on a fluorogenic substrate and it releases fluorescence, and so that's the signal that we measure, but notably missing because it is formulated particularly to do this, there is no factor G pathway. And even in the recombinant cascade reagents, we have the three major parts of the cascade, the factor C, the factor B, the pro-clotting enzyme, but notice again, there is no factor G. And so the good news about these recombinant reagents in the variety that they come from is that they are deliberately made not to have that factor C component. And so any kind of an interference, a so-called false positive that we used to see in the past, is now all eliminated. So right now we currently have two types of recombinant reagents. We have recombinant cascade and recombinant factor C. We just talked about that. What's interesting right now is that these two types of reagents come from three different genera of horseshoe crabs, Carcinoscorpius, Tachypleus, and Limulus. And the proteins are expressed in a variety of mammalian and non-mammalian systems. So now we've got a mix and match of the actual source of these proteins and the expression systems that generate these proteins. They are further formulated for stability. There is no factor G pathway, which is a good thing. Now, as far as USP goes, it sees the recombinant methods as alternative methods to 85 being the Bacterial Endotoxins Test. And right now, I have to say that our new chapter, which is called Chapter 1085.1, which describes the USP's thinking with respect to recombinant reagents, it is out right now for comment. So if you don't know that it's out for comment, please take some time, go off to the USP website, take a read, and send us back some comments. Now, per the USP general notices, alternative methods need to have the following three components. They need to have analytical comparability. That means we need to be able to make a linear standard curve and, all of those sorts of things that go along with it, ruggedness, reproducibility, all of that stuff. We have to be able to demonstrate suitability, which is inhibition enhancement, as we see in the 85. That's part of the current 85. The one that we struggle with right now is a test result comparability. Okay, what is that? That's the autochthonous endotoxins. In real life, do the recombinant reagents recover autochthonous activity to the same degree that the current compendial ways recover that same activity? Now, this one, I wanted to show you this, not because it's relevant in its philosophy to what we're looking at, although it was done very, very long ago with respect to the rabbit pyrogen test when it was being compared to the LAL test. And these were done by the people in Baxter. And what you can see down here is that the E. coli activity was measured, E. coli equivalence. What they did was measured autochthonous activity, and because the reagent was standardized back to E. coli, these are the E. coli equivalents over here. And over here is the rabbit test. Okay, and so these were the rabbit tests and the increase in temperature. So down here, this quadrant said we had agreement that we had low endotoxins and low fever, so that's good. This quadrant up here would say we have agreement because we have high endotoxins and a corresponding high rabbit fever, and that's good. You can see all of these dots over here. That's good. This quadrant up here basically would say, okay, we would have a false negative LAL test, meaning that the LAL test found little or no endotoxins activity, but the rabbit found a lot. And look at this one is empty. So in this particular study, there were no so-called false negative LAL tests with respect to what the gold standard was at the time, which was the rabbit pyrogen test. And that's a good thing. In this quadrant over here, okay, what this quadrant says is that we found a bunch of LAL activity, but the rabbits were negative, meaning the rabbits didn't respond, but we got LAL tests responding. And as you can see, these might be called false positives. These might be called the fact that the LAL test is more sensitive than the rabbit pyrogen test, but there are a few who fell in here. That might even be glucan for all we know. This was done on human serum albumin, 125 lots, as I believe. And so these were real products contaminated with true autochthonous endotoxins. In terms of comparability, right now, data out in the public domain, there's a great paper by Dr. Kikuchi and his colleagues published in 2017, and that's on the list of references at the back. But you can see what he did here is he took and he compared a whole bunch of different LAL BET tests. I can't say they're all LAL. Endospecies is an interesting one because it's a TAL test. It's a tachopleus lysate. But what they did here on this one is they fractionated out the factor G pathway and then put the rest of it back together. So what you have here is a reagent, a naturally derived reagent from which the factor G pathway has been taken out. ES2 is an interesting one. It's an LAL reagent currently made by Fujifilm, Wako, and this one is an LAL reagent to which a glucan blocker has been added. The kinetic KQCL is a product that we all know is a Lonza product, a current compendial, fits the compendial mold product. PyroSmart is the associates of Cape Cod Segugaku. Cascade reagent, pyrogene, and endozyme are each recombinant factor C reagents by Lonza and bumeriu, respectively. And so what Kikuchi did was he said, okay, I'm going to measure, okay, at a number of laboratories, we're going to measure the responses of all of these BET reagents, and we're going to compare them. And from these data, you can compare all kinds of things. You can compare the cascade reagents to the non-cascade reagents. You compare all of these reagents to the current compendial method. You can compare all of these to what's a normal TAL reagent that doesn't have a factor G pathway. And so you can look at these data and you can compare all kinds of things. And so if you don't have a copy of this paper, I would suggest that you go ahead and do it. These 5 materials up top were so-called NOEs. These were made in the laboratory. These down here are in Japanese, but they represent various pond samples, river samples. One of these is even a household sewage sample, but these are samples that one is a mineral water sample. I don't remember which one is which right now. I have that translated somewhere, but the point is that these are all materials that you would find out in nature that are likely to contain endotoxins from autochthonous organisms. Endotoxins are only found in the water system. Well, that's false. While most organisms might be found in the water system, and you figure the oligotrophic environment to which these organisms adapt is really the water system, right? Purified water has a very low nutrient content, and so that's a good place to find these autochthonous organisms in a pharmaceutical manufacturing facility. But sometimes you can get these organisms or endotoxins coming in, particularly if you use raw materials from some sort of a natural source. I've worked with bovine and porcine heparin full of endotoxins. That comes from cows and pigs. Diatomaceous earth is full of endotoxins. Sometimes, depending on the grade that you get, sugars, mannitol and dextrose can be full of endotoxins. And they or may not, they may be from the same organisms, but they've certainly adapted to a different environment than organisms that we might find in the water system. So while I would go and say that predominantly organisms that may find their way into your product are likely to come from your water system. If you do have in the formulation of your product anything that comes from a natural source, it also is likely to bring in some endotoxins along with it. Again, these are autochthonous endotoxins, not the endotoxin standard. Another thing that maybe people don't get, most endotoxins testing is done on finished product. Well, actually, that's not true. Most of the endotoxins testing is done on water systems. In fact, I've heard that up to 50% of all endotoxins testing done in the world these days is done on water systems, and certainly then probably comes raw materials and in-process testing. When you think about it, relatively few endotoxin tests are relegated to end product testing. What's the minimum, right? A beginning and a middle and an end sample that you can test individually or that you can pool. And when you think about that, number one, what a statistically invalid sample size given the large sizes of most, certainly most biological and small volume parenteral manufacturing lot sizes. 3 vials is a minuscule number. So very, very little, relatively speaking, very, very little is spent on the finished product testing, which is a good thing because it tells us that what we're doing is we're doing the right thing by GMP. We're controlling all of the stuff in the middle to make sure that this stuff in the end is clean. So I want to talk a little bit about endotoxins control because I think this is one of the important intended uses of BET. So we're going to take this. I see this as a three-sided event. The first thing is to establish control of your process. The next thing is to maintain control, and the next thing is to monitor for continued control. So establishing control, what does that mean? Well, you validate stuff. You validate the water system. You validate the depyrogenation process. You validate the cleaning process. If you look back on FDA sites from a number of years ago, okay, you can see that many of these citations are coming from the fact that there's standing water, that the cleaning validations were inadequate, that the preventive maintenance was inadequate, that stuff wasn't properly validated. And you can see that a lot of the problems, endotoxin problems, really do stem from the fact that there is a breach in the establishment of control. You want to make sure that all of the materials are dry. Anything that you wash is dry because anything that has puddles in it, gram-negative bacteria can grow. If you buy if you buy your materials from sources, you want to do a proper vendor qualification and also enter everything into a preventive maintenance program. And once all that's done, how do we maintain control? Well, we have SOPs, we have training. Change control is hugely important because if something changes, okay, that generally, or not generally, may often, is the cause of a pyrogen problem. That something changed and nobody told anybody, and all of a sudden we have an endotoxin problem. And sometimes the smallest changes can result in very big problems, hugely important. The CAPA system, looking for the sources of endotoxin contamination, is hugely important. Calibration in PM, and again, vendor management, hugely important. Then we look to monitor for control. We have to understand that monitoring is not a control mechanism. Everybody wants to say that monitoring is control. It isn't. What monitoring does is it's but one way to assure that all of the controls that have been established are working properly. So our monitoring is not a control mechanism, it's really a checking mechanism. It's a surveillance mechanism. And when our monitoring starts to go out of control, if we see an adverse trend, then we can go back and start asking questions to say, what's going wrong with our establishment or maintenance of control that's causing these trends to go up, and hopefully we can clear that up before something fails. So if you look here in a typical 10,000-foot McCullough simplified way of doing things, you have incoming raw materials. Those things get formulated in aseptic operation. They might get filtered through a 0.2 micron filter. There's a fill finish, and there's laboratory testing. And so my challenge to you guys is to look at your process, a whole lot more complicated than this one, and use any kind of a risk assessment that you like in order to determine the critical control points. Critical control points meaning those places in the process where endotoxins can enter, but also places that we've put in there to make sure that we reduce or eliminate endotoxins. And so we look for critical control points. My favorite one to do is HACCP, okay, which is, there's a really good tutorial on that on FDA's website, but you can use any one that you want. And it's always good to do these things in a group of people because one person usually has some sort of a bias. And so, of course, I did this all by myself. So we have to take it with a grain of salt. Well, the process step, what about raw materials, receipt, and testing? Yeah, that could be a critical control point, particularly if our materials are derived from natural sources or non-GMP manufacturers. Now, this becomes really important for current cell and gene manufacturers very often. We buy vectors, we buy plasmids, and we buy these things from places that may not have the same historical level of GMP control that we are used to seeing. So those things can come in with endotoxins in them. So what do? Well, we confirm the C of A. Sometimes we test, sometimes we don't. We monitor this. We do maybe incoming skip testing, but we recognize which ones of these raw materials are potentially problematic and which ones aren't. What about formulation? Well, yeah, that's critical because of the water for injection. And then we have to make sure that the vessels, that there's no cracks in the vessels, that the vessels are clean, that they're held in a dry state, all of these things. So yeah, formulation becomes pretty critical. So we monitor the water system frequently. We have cleaning validations. We do bulk and in-process testing for endotoxins at certain points in the process, which you will identify through the risk assessment. What about aseptic filtration? Well, actually not really. That's not a hugely big control point for endotoxins because endotoxins in the form of outer membrane vesicles and in the form of little piece parts of cell wall fragments that come from the death of these microorganisms go smack right through a 0.22 micron filter. So while they might take out whole live bacteria, they don't take out endotoxins. They're not meant to do that. So that really doesn't become an issue in terms of monitoring. What about fill finish? Yeah, the container closures become an issue. But you know what? Once you get it validated, you don't have to routinely test that. What is depyrogenation of vials? Okay, we validate the depyrogen of vials using endotoxin indicators, using endotoxins, but really it's a function of time and temperature. The laboratory testing, I think, is probably the most critical step and probably the most overlooked step. If the laboratory doesn't generate accurate data, then none of the rest of this matters. So for the McCullough Pharmaceutical Company, if there were such a place, one of my big focuses would be on the laboratory. So in terms of summary, the use of the BET assays, we really want to use them to look for autochthonous endotoxins, endotoxins from autochthonous bacteria. The endotoxin and pyrogen technology is advancing. Okay, we now have recombinant methods. We have new methods and instrumentation that use less lysate. Where monocyte activation test is on the horizon. And remember, for any of these, what we need to make sure is that we understand the three requirements: analytical comparability, suitability, and test result comparability. And I think that's it. Back to you, Marcus.
Thank you very much, Karen. It was a great presentation. Now, before I introduce our next presenter, Veronica, once again, I would like to remind our viewers that there will be a live question and answer session at the end of Veronica's presentation. So please feel free to send your questions at any time during the webinar via the questions tab, and that's located directly below your webinar screen, and we'll go through these at the end. And please also state in your questions which presenters they are for. And now please allow me to introduce our second presenter. Veronica manages the Technical Services Group at Associates of Cape Cod. She joined the team in 2007, and thanks to her strong knowledge of biochemistry, microbiology, and immunology, She brings an expertise that is vital to Associates of Cape Cod's customers when it comes to technical support of testing complex samples, low endotoxin recovery matrices, and troubleshooting of bacterial endotoxin tests at all levels. Veronica speaks frequently at scientific conferences and seminars globally and is established expert on endotoxin testing. Veronica holds a master's degree in biochemical engineering from the Institute of Chemical Technology in Prague, Czech Republic. And now I'll be handing over to our second presenter. So welcome, Veronica.
Thank you, Marcus. Good morning, America. Good afternoon, Europe. And good evening, Asia. Greetings from Falmouth, Massachusetts. Before I start my portion of this webinar, I just want to mention that the information provided in this webinar is for discussion and educational purposes only, and that any decisions and actions should be based on relevant regulations, not on this presentation. I'm going to go quickly through the humble beginnings of the endotoxin test, the LAL, ACC's role in that process. I'll touch on manufacturing quality, sustainability, and innovation. Karen provided me with a very nice overview of what the endotoxin test, the LAL test, is intended for, and I hopefully can now contribute a little bit more details as well. Did you know that the LAL test was discovered in this little seaside village that you can see on the left side? The village is so small that it's called a hole. It's called Woods Hole. And we're now looking at the marine biological lab where Dr. Bang and Dr. Levin discovered the test back in 1964. Now, I did not know that when I first moved into this area, and when I started discovering the streets of Woods Hole, and I discovered what a hidden gem Woods Hole is, I happened to just simply walk into an MBL gift shop just to buy a stuffed toy for my toddler at the time, who is now 15. And that's where I ran into this, I call it the Wall of Fame, and which is lined with various plaques that commemorate the different discoveries by both MBL and the Woods Hole Oceanographic Institute. And I have to say that's where I actually learned for the first time of the now infamous discovery by, as you can see here, Dr. Bang and Dr. Levin. So fast forward just about a year later when I joined ACC, I learned a little bit more about endotoxin and about the history of it as well, some of which I would like to share with you today, because sharing and appreciating history is very important. So as you can see here on this timeline, only about 9 short years later, the FDA published the first manufacturing standard for manufacture of LAL, because it was recognized that LAL as a preparation is it will have some very significant utility in the measurement of endotoxin at the time, primarily in pharmaceutical products, because their testing by the rapid method was somewhat difficult. Dr. Stanley Watson, who was at the time the Woods Hole Oceanographic Institute senior scientist, and he was investigating the use of the horseshoe crab extract as a way to measure bacterial biomass in seawater. He was preparing various preparations of the LAL reagent, and the preparations became requested by some pharmaceutical companies. So he decided to found Associates of Cape Cod in 1974, and after FDA published the final manufacturing standards for the LAL reagent in 1974, Associates of Cape Cod was the first company who was granted the FDA license to manufacture LAL. It's a license number 700, and we continue to manufacture LAL under this license to this day. Later on, FDA also granted us the license for manufacturing of other lysates as well for all of the 3 methods, as we now know as gel clot, turbidimetric, and chromogenic. And throughout the early 80s, that was the time when the LAL test has been basically subjected to the comparability study, as Karen was mentioning, to compare it to the predicate method, being the rabbit pyrogen test. So there was a lot of time and a lot of effort that was put into to show how LAL really compares and whether we can rely on the LAL results. So to this day, FDA, it's the Division of the Biological Standards and Quality Controls. It continues to oversee the LAL production, and that for us, for the end user, assures quality and proper certification of the LAL reagent. So I think it's really safe to say that the LAL test and the quality of the LAL reagents for sure elevated the quality and the safety of the drug products, including vaccines, including medical devices that we all became accustomed to and we continue to rely on. The way that ACC is involved in this is that we continue to build on this expertise, this history, this knowledge, and as the global endotoxin and glucan experts, and we continue to manufacture the compendial LAL reagents for the past 45 years, or a little bit of over that. We also have taken on sustainability projects. Since 2011, we have a growing commitment to sustainability, and we specifically partner with suppliers that share this commitment with us so that we can collectively reduce our environmental footprint of our operations. That is of great importance to us. One of the projects that we took on is the wastewater reduction and reuse project, where we recycle wastewater locally. It is multi-purposed for cooling of gases and recycling processes. Another one is recyclable product packaging. Another way of doing so is the use of more environmental friendly materials, glues and inks and so forth. There is one project that we are particularly proud of, and that is our unique horseshoe crab sustainability project. Under this, we so far released nearly 750,000 juvenile horseshoe crabs back into their natural environment. And this project was started only a short 2 years ago, and recently was also expanded into Asia, where horseshoe crabs are considered to be endangered species. What this means is that we, under this program, use eggs collected from horseshoe crabs that are going to go to bait industry. And so these eggs are being collected, they are being fertilized, and then they are being grown out. So the embryos are then being grown under the specific conditions. And then later on, when they become juvenile and strong enough, we introduce them back into their natural environment. This all happens under a Class 1 Type 4 aquaculture permit, and we also have a patent pending on this. And so we hope that this will help to ensure the stable supply of horseshoe crabs now and for future generations to come. Another sustainability effort that we are very proud of to be part of is the development of a sustainable reagent using the recombinant technology, as Karen already mentioned. This is done in cooperation with our parent company, Seika Gakko, and in early 2010, Seika Gakko developed recombinant clones based on the genetic sequence of Tachypleus tridentanus, which was formulated into a recombinant cascade reagent, and this then became commercially available on the Japanese market in 2015 and has been since widely used in Japan and has been published on. It was also included, as you saw, in the Kikuchi study. So how does that exactly compare to the compendial LAL reagent? Again, Karen did a great job explaining that to us already in great detail, so I'm just going to summarize here. Here we're looking at side by side what is inside the recombinant cascade reagent in the middle, and on the right what's in the LAL reagent. So the big difference is that while they both do contain the enzymes that are required for the cascade mechanism for detection of endotoxin, the LAL reagent also contains other components of the amoebocytes that are extracted with, and a lot of them have various roles, for example, upregulating the coagulation mechanism, and of course the natural substrate coagulation is in there as well, along with the factor G enzyme, which has been widely shown to be obviously causing the enhancement or co-reactivity with glucan. So if we have a sample that contains endotoxin and glucan, the LAL reaction is likely to produce enhanced results because of the synergistic effect between endotoxin and glucan. That, again, as Karen said, is not the case with the recombinant cascade reagent because the recombinant factor G was not cloned, it was not added in there. The rest of it, really, between the reagents is the same. The reaction, the mechanism, the principle is identical. So these reagents are homogeneous in the way that they react and in the way what input, or I should say output, they produce, as both of them are chromogenic, kinetic chromogenic reagents, and they both rely on the use of a chromogenic substrate that you can see here. The regulatory landscape around the use of recombinant reagent is a dynamic landscape for sure. Karen was already explaining to us the USP's thinking on the recombinant reagents, that they consider them to be alternative methods. And here you have the full reference to the new draft in revision text of the chapter 1085.1, which is called The Use of Recombinant Reagents in Bacterial Endotoxins Test, Photometric and Fluorometric Methods Using Recombinantly Derived Reagents. The deadline on the comments is November 30th, so if this is of interest, it's definitely a good time to read through that and submit those comments. The JP also considers the recombinant reagents to be alternative. We expect that they will be including them in a general chapter that should be published in Japanese Pharmacopoeia 18th edition, scheduled for April 2021. The European Pharmacopoeia moved forward with implementation of a new compendial chapter 2.6.32 named Test for BET Using Recombinant Factor C. This chapter includes the recombinant factor C reagents only, and it will become effective on January 1st, 2021. So from this, it is clear that the pharmacopoeias are not harmonized in their thinking and their understanding of the recombinant methods right now, but this is a developing discussion and developing situation, and we continue to monitor that situation as well. So what does it exactly mean when we say compendial versus alternative? This is just a little summary of what Karen was explaining to us earlier as well. So any compendial test is a test that is considered to be validated already. The validation was done part of the manufacturing, so by the manufacturer already, or and by it was accepted by the experts in the industry. It also means that there has been already plenty of data showing the comparability of that method to a benchmark to a predicate method. The chapter 1225, named The Validation of Compendial Procedures, outlines exactly how the method validation has to happen. So obviously, on the other side, the alternative test is not considered to be validated yet. So that means that the end user would have to go through the method validation process, and Again, as Karen pointed out, the alternative test is alternative because it is not considered to have been shown enough data that showed the comparability to the predicate method. So the comparability of the testing still remains to be answered when it comes to recombinant reagents. I will touch base a little bit here on the analytical performance, but also on advantages that the recombinant reagents may be able to provide to us. And this is specific to the recombinant cascade reagents. Because if there were no advantages, why would we even bother with doing the extra work with implementing them? You can see here in green, linearity is the big deal because linearity of the standard curve is of the utmost importance, and I will touch base on that later on a little bit. The speed of the test is very important. So how long is my test to result? Can I get my results faster? Yes, you can with the recombinant cascade reagents. What is the lot-to-lot consistency? Definitely way better than for the LAL reagent, because naturally with the recombinant cascade reagent being engineered, we can monitor, we have more tools at our disposal to monitor the lot-to-lot consistency. And again, the absence of cross-reactivity with beta-glucan is of huge interest to the recombinant reagents as well. So suitability to various types of products is obviously one of the requirements. So if the reagent does not work for a wide range of various products, then there would be no point of using it. So let's take a look at what we mean by the linearity. I picked it here actually to talk about it a little bit more because it's fairly easy to explain. Most of us are very familiar with the pharmacopoeial requirement for the linearity of the standard curve, the fact that the correlation coefficient has to be at least 0.980, and there's also additional criteria or additional references in the USB chapter 1085. That's a newish chapter called the Guidelines on the Endotoxin Test. And that describes the standard curve control, because there's many factors that go into the bias of the method. There's many factors that actually will be responsible for a lower linearity. The reagent could be one of them for sure. And so clearly, if we are running a standard curve, the curve really does have, for an LAL test, it will have a curvage to it. Our job is to make that curvage as little as possible. So on this slide, you can see that the curve that was measured had an R value of 0.981, which is still technically valid. However, I would not consider it exactly a great curve, because now the points, as they're fitted with the linear regression, the actual points are actually going to be quite far away from the straight line fitted through it. So our job is to make, or to attempt to yield, an R value as close to 1 as possible. That is the best outcome. The linearity directly impacts accuracy of my testing. So the lower the linearity, the lower accuracy. So here you can see that based on 2 publications that were looking at the reproducibility and linearity of the recombinant cascade reagents, they showed that the linearity was exceptional and it was repeatable. On the left, in Kikuchi's study, and this is from 2018, so that's a different paper, 3 lots of the recombinant cascade reagents were analyzed. Each lot was analyzed 15 times, so it means these are 15 individual runs. And the R value, the mean R value for those 15 runs was either 1 or 0.999, which is really, really high, and it's basically, as best as it could be. And on the right side, you can see a publication from Moroi from last year, where the number of runs was 12 with different users, so 2 different technicians. I believe those were 3 different lots of the recombinant cascade reagent. And so the 12 runs, their mean R value was 1. So obviously that's perfection. And this is for a wide range standard curve from 50 down to 0.05. So why does it really matter, what my linearity is? I mean, obviously, if I can fit my curve above 0.98, I should be good to go. So hopefully this slide will demonstrate that a little bit better, basically the relationship between the linearity and then the accuracy over test. Of the test. So the red line here shows the recombinant cascade reagents R value, so the correlation coefficient value of 0.99. And so if we use the slope and the y-intercept of this standard curve to back calculate the measured endotoxin concentration of the standard, and if we normalize it against the nominal values, times 100%. You can see here that the red line is basically hovering around 100%. So what it means is that I'm measuring for every concentration, 50 to 0.05, I'm getting around 100% for each one of them. So my accuracy is extremely high because I'm hovering around 100%. And that's the blue line there, if my R value drops down to 0.997, which is still an amazing curve, as measured by the LAL reagent, regular LAL reagent, you can see that the back-calculated values, their accuracy does change now. Now I basically have a variability of 20%, the middle of the standard curve. And that is because of the curvature of the curve, because of the bowing. So the middle of the standard curve now produces the accuracy that is off by 20%. And so it's overestimated. And it's the middle of the curve where we actually always place our spike samples. And we calculate the spike recovery based on that. So that will always actually come out slightly better just because of the curvature of the curve. And on the edges of the curve, we actually have underestimation because we're getting only about 80% back of what we put into the test. So again, the more linear the curve is, the better our accuracy is. I mean, this is all well within the 50 to 200% accuracy of the test. So it's both of these tests are perfectly valid, but it is always desirable to improve the test and to be able to control it as much as possible. Another great way of showing how the recombinant cascade reagent is suitable for testing is this suitability study, which was also published. It was a very large study including 119 different final drug products, and they were tested at various dilutions. And the way that the data was interpreted was by looking at the non-interfering dilution. So what is the lowest dilution possible in order to get a valid result? And so this usually indicates an interference pattern that we can see from the, coming from the drug product. In about 50% of the drugs tested, that non-interfering dilution for the recombinant reagents was lower than for the LAL reagents. So that means that the recombinant reagents allowed us to test it at a lower dilution, which is always desirable because with every dilution, we may lose the visibility of the endotoxin. In 8% of the drugs, the dilution had to be higher by the recombinant reagents, but that was associated primarily with samples that actually preferred to be tested by a turbidimetric assay versus chromogenic assay. The remaining of the drugs had just about the same interference profile. So if anything, we can say that the recombinant reagent performed the same as, so it is at least equal to the LAL reagent for testing of wide range of final drug products. We continue to test more types of products, those that also contain a large background of protein, because protein is certainly one of the factors that can cause interference, both enhancement as well as inhibition. So here I have actually very new data that hasn't been published yet, and this is a comparison side-by-side ALL test versus the recombinant cascade reagents, all tested at exactly the same time on the same plate at exactly the same dilutions. And you can clearly see the differences in the PPC recovery, that with absolutely no intervention, the recovery improves just because I used a different reagent. In most of the case, I mean, it improves in every case. And in one case, that dilution of albumin was not high enough for the recombinant reagent either. None of these, unfortunately, contained the indigenous endotoxin that Karen was talking about. So we are not getting information from here on the comparability in terms of detectability of the indigenous endotoxin, which is, by the way, still the issue, or it continues to be under evaluation, and it continues to be a subject of discussions. We all want to be comfortable with the fact that the recombinant reagents will be detecting the same amount of the indigenous endotoxin as the LAL reagents. Just to document how dynamic this conversation is, lately, actually in August, there were 2 publications that came out. Both of them are review papers, the one by PDA, published in the Journal of Pharmaceutical Science and Technology, and it is called The Currently Available Recombinant Alternatives to Horseshoe Crab Blood Lysates. And another review paper was published in the APR, so the American Pharmaceutical Review, and it's actually a 2-part paper. So the part 2 was published recently, and it specifically speaks to comparability. There's a lot of great information and review of the data that is available in the public domain. So for those who are interested, this is a great read, and it provides, along with the 1085.1, it provides actually a great sort of setup for future studies. So another thing that we also have to be aware of in terms of the recombinant reagent is the oversight of their manufacture, or to be more exact, it's the lack of the oversight of their manufacture. So currently there is not a regulated agency or regulated authority that would provide the oversight over the manufacturing quality of them. So what this means for the end user is that the end user really has to be now more careful and more thorough in the process of auditing the manufacturer. So because the manufacturer of any reagents for endotoxin tests would be considered a critical supplier. And so the supplier audit is going to be a critical piece of this, and it now solely relies on the end user, on the laboratory user, because there is no licensing, no FDA licensing for this. So there is no automatic assurance that does lie with the LAL reagents. So again, the recombinant reagents and LAL reagents should not be considered to be interchangeable. They are different entities. The methods may be homogeneous, but the entity is different, and that's how it has to be looked at. And like I said, there is a lot of really great information in the 1085.1, which shows the current thinking on this topic. So when it comes to this innovation, which is very exciting, we generally, as human beings, I think we have a tendency to go for the new thing out there, but we also have to pace ourselves. We do have to make the right choice for the right reasons. And one of the right reasons, obviously, is this reagent the best type of reagent for my product? Because every product does behave differently. Speaking of testing the final product. does the manufacturing quality and the oversight of it matters to me? What are the studies? What are the studies telling me about the reagent itself? We also have to consider the expertise, longevity, and the sustainability efforts of the supplier. So all of that's a part of a big picture. And ACC is very happy to be part of that big picture. We are really proud to continue to develop and to take the endotoxin testing to the next level as well. But I think that throughout the presentation of Karen and myself, hopefully we were able to show that the endotoxin test as intended to be used has really shown how critical it is for the monitoring and for the routine testing of our drug products, and it ensures the quality of the drug products, including many, vaccines, and now everyone talks about COVID vaccines, so that is included in that picture as well. The innovation is pushing in. The recombinant reagents are earning its place under the sun. There is still some work to be done. There is still comparability data to be shown, and the pharmacopoeias are not harmonized just yet, but these are very exciting times, and we're happy to be part of that process. And with that, I would just want to say, if you have any questions, if you are looking for any educational content, we have great content on our website at acciusa.com. You can also follow us on LinkedIn, and you can always send us any questions to techservice@acciusa.com. Thank you for your attention, and over to Marcus.
Thanks a lot for that, Veronica. And welcome back to the screen, both of you. So it is time now then to begin our Q&A session. But before we do, once again, audience members, you can still send your questions via the questions tab, which is located directly below your webinar screen, and we should get through to those shortly. Please also state in your questions which presenters they are for. And then our first question then, I will address to Karen. Hello there, Karen. So question number one, Karen, do B-glucan blockers block all glucan activity with regard to endotoxin testing?
No, the literature would suggest that there is a spectrum, a continuum of different kinds of glucans, and also the effectiveness of the glucan blockers. And so, that is one issue that still has to be worked out by every user. I would suggest if you are using an LAL reagent and a glucan blocker, that you talk with your manufacturer who supplies the reagent and the blocker, because they have the most experience with it. But it's not necessarily a given that all glucan blockers are 100% effective against all glucans. I don't know. What do you think, Monica?
Yeah, I agree with that. Definitely. The subject of glucans is not a trivial subject. Glucans happen to be very complex, heterogeneous substances. And so just blocking them basically with one generic buffer is sometimes may not exactly, because it's a biological test as well, so it may not exactly translate. So the glucans are definitely one variable in the comparability between the recombinant and the LAL reagent that do play a role.
Thanks a lot for that, both of you. So our next question, Karen. Can endotoxin remain viable in the acids, alkali, and or organic solvents?
It depends. Okay, if you read the literature, endotoxins, and most of this work has been done on purified endotoxins, but endotoxins are certainly much more susceptible to alkali than they are to acids. The literature would suggest that if you have acids combined with heat, that's important. It depends on the normality of the acid or the base. So I think the answer is, unfortunately, it depends. And I don't know that we can make a one way or another answer to that. And like I say, most of the testing has been done on purified endotoxins in the end. So I'm not really quite sure if there's a lot of literature out there on autochthonous endotoxins with acid, base, and organic solvents.
Great, thanks a lot. Before we move on to the next question, Veronica, is there anything you'd like to add to that answer?
No, that was great. Thanks, Karen.
Okay then. Our next question, Karen, once again. After an endotoxin OOS for a medical device, additional investigation testing is performed. Can you resample and retest be performed using a larger sample size? And the initial OOS results be either confirmed or, if not, then retested results averaged with the initial OSS results?
Well, kind of a loaded question. Okay, so I think that we have to look back when the original, I think it was the original guidance document, 1987, allowed for 2 retests. The first retest could be of the failed, let's say, a pool of things that you did. The second retest was 10 individual units. Well, all of that is gone now. And we have to look back and we have to understand the premise and the philosophy of the OOS guidance, which means that you can't test something into compliance. So I would say that, number one, there are no automatic retests. You need to find out first why the first one failed. You can do investigative testing, which may include some sort of tinkering with your tests so that you have a hypothesis about why the first one failed. But you just can't immediately go to a retest and decide that I'm going to retest this before I do the investigation. So I think that you have to be very, very careful about that. And in the end, if you find a reason why that initial test was no good, you invalidate that original test in your investigation. You say that original test was no good because we found evidence of X and Y and Z, and therefore it is an invalid test. And so the only test results that you report, even though you keep all of them in the data set, the only ones you report are the ones from the new test that is a valid one. So I think that we have to be really, really careful about this idea of doing another test and doing another test in order to prove that the first one is wrong. Additional testing doesn't prove that the first one is wrong. You need to understand why the first one was wrong.
Thanks a lot for that, Karen. Before we move on, anything you'd like to add to that, Veronica?
I think that was exhaustive.
Our next question is, Karen. Yep. There are no CV requirements for kinetic chromogenic assays. What would be an acceptable CV percentage to test?
Can I take that?
Yeah, of course. Go ahead.
Okay, so that's true. There is no regulatory requirement for CVs, which stands for correlation of variation. However, it kind of became a industry requirement. It's probably a strong word. Maybe I should say industry sort of guidance, or industry standard that everyone does monitor CVs as a measure of repeatability, because it is a really useful number to take a look at very quickly. So what is that number? That number depends on what population we're calculating the CV from, because it's actually a statistical tool that we're using. So generally speaking, if we're calculating CVs based on onset time is a large number, so the CVs, because that they're relative numbers, they really should be then smaller. They usually up to 10%, which is what I've seen in the industry. And if we're calculating the CVs based on concentration is generally a small number, and so the relative values for the CVs are going to be larger. They go maybe to 25%, generally speaking. But this is usually added to the SOPs, and so then it becomes a in-house requirement.
Okay, thanks a lot for that. Anything Karen would like to add to that?
Yeah, I wonder about the utility of the CV when you only have duplicate samples. So if you have duplicate samples and they don't match because their CV is greater than N%, now you're stuck with, well, which one of these is correct and which one isn't, or are neither one of these correct? And so I think that the sample size, the fact that we do LAL testing in duplicate, really does restrict the utility of the whole concept of CV. If they're close, it'll tell you, if the CV is good, it'll tell you that that's a good thing, that you got your answers are close. If it's wide, I'm not sure what that tells you.
Great. Thanks a lot for that. Our next question, I'll address to Karen, but feel free to jump in, Veronica, if you'd like to. What is the acceptable BET level required in raw materials for an injectable formulation?
Well, so the answer to that is it depends, right? You have to look. So for every particular parenteral formulation, okay, there's an endotoxin limit that's calculated for that formulation. And so what you need to do is you need to say, okay, here's what I'm allowed to have in the final formulation. And you have to go backwards and say, okay, here are the various components that are part of that formulation. And you have to assign internally based on the component and based on how much of the component is in your formulation. You basically are charged with assigning all of the endotoxin limits to the various components, such that if they were all at their limit, they wouldn't exceed the endotoxin limit for the final product. So all of that really falls on the user, and it depends on the product, its endotoxin limits, and its formulation components. Now, what happens is if you have a number of products that all have the same component, let's say you have 10 products in your company and all the products have sodium chloride, well, then what I would do is calculate all of the varying contributions that are allowed by sodium chloride and choose the most stringent one. Because if you know that it works in the most stringent capacity, it'll work in all of the other ones. But you still have to go back and figure out each one of these products on its own. There's no special algorithm to do it.
Great, thanks a lot. Anything Veronica would like to add to that?
No, there's nothing to add to the greatest answer that Karen always provides.
Great. Okay, moving on then. Our next question is for Veronica. Where's it gone? Just lost it. 1 second. What is the survival rate of juvenile crabs released? Do you have any data about this point?
Okay, that's a great question. So we wish we had data on that. We expect that the juvenile crabs, and they're still very small at the time of the release, so you can't exactly tag them. That is impossible. But the expectation is that the survival rate is the same as the survival rate of any juvenile horseshoe crab. So whatever happens in the nature is what these little babies made by us, but also subjected to. So we hope that, if it's 10%, then now we are going to have 75,000 more horseshoe crabs in the ocean. So it's always a good thing. But yeah, there's not an easy way to really collect the data because of their size at the time and because they're going back into their natural environment and they float away into, burrow themselves into the sand probably. Yep.
Great.
Veronica. It's really great.
Mm-hmm.
Okay, our next question then, Veronica. What about China pH, about recumbent methods?
Chinese pharmacopoeia, yes. I neglected that to include it in the slide. So they also actually included it's a general chapter on recombinant factor C reagents. So recombinant factor C reagents are in the Chinese pharmacopoeia. Yep.
Great, thanks a lot. Before we move on, anything Karen would like to add to that answer?
No.
Okay. Next question, Veronica. What brand and type of LAL do you use for linearity and interface comparisons?
Oh, in the comparison that I had on my presentation, it was actually a kinetic chromogenic reagent. So the R value for the LAL reagent was a typical R value that you're going to get with a kinetic chromogenic reagent.
Thanks a lot for that. Anything Karen would like to add to that?
No, I think it's a great question. I think people need to remember as well that the LAL, the cascade recombinant reagents, are both, to my knowledge, Veronica, kinetic chromogenic reagents. And we need to understand that the recombinant factor C ones are endpoint tests. And so they're not analyzed in the same way. Okay, so if you want to draw the analogy, the recombinant factor C ones are analyzed more like the endpoint chromogenic currently in the pharmacopoeia. Okay, and so it's a little bit different.
Great, thanks a lot for that. Our next question. Does ACC have comparability data for their fully recombinant TAL? Veronica.
Yes. So there has been, so some of the data has been published, part of the Kikuchi study. So when we speak of the comparability data, I think the discussion moved into the field of speaking specifically about the detectability of the indigenous endotoxin. I can say
Goodness, I know it took me a long time to figure
I'm
that
crying. Same thing. So the only problem with that is that there is not that many products that will have detectable endotoxin in them. So I think that is really the biggest hurdle. So I personally am constantly on a hunt for samples for real manufactured drug products, medical device products that have detectable endotoxin in them. Some studies went towards looking at water and the endotoxin, indigenous endotoxin in the water, because that's where we do find it quite easily. So yes, we do have that is our, major focus, because I understand that is the missing piece sort of in this recombinant world. We actually presented some of the data in webinars. We have not put it in a one publication, yes, but it is on the way, working on it. So, yeah.
Anything Karen would like to add to that before we move on?
No, I think that's great. And Veronica's right. I mean, over the last 40 years, the LAL test, by its intended use, has helped us to clean up our act. And so in terms of final products, we don't find nearly as many contaminated products as we did. But that doesn't mean that there aren't any. And, there are definitely instances where we know that because of a breach in process or a breach in PM or some issue with the water system, that we can find these things. You can go backwards. A lot of times you can go backwards in your process and look at in-process materials that may be contaminated with endotoxins. Maybe you have an endotoxin removal step somewhere, but upstream of your finished product, perhaps you have a place where there are autochthonous endotoxins that can be measured. Certainly water, deionized water has been shown, upstream of any still or upstream of any RO membrane has been shown very often to have measurable autochthonous endotoxins. So I think that there's places we can look, but Veronica is absolutely correct. The opportunity to find these things in finished products is pretty limited.
Thanks a lot for that, Karen. Our next question for Veronica. What endotoxin separating process are the best ones not to damage the animal source ingredients?
I'm sorry, can you repeat that question?
Yeah. What endotoxin separating process are the best ones to not damage the animal source ingredients?
Animal source ingredients.
Animal source ingredients.
We're talking about depyrogenation of something?
I'm not sure.
No problem.
So I can offer answering this question offline. If you just email me at techservice@ACCI-USA, and we can bounce back and forth to make sure that I understand the question. Sorry.
Okay then. So moving on to our next question. There is evidence now that B-glucans in natural waters can cause an increase in activity. What are your thoughts on this? Veronica, would you like to say this?
Sure. That they can cause increased activity. I mean, yes, beta-glucans in any matrix can do that. And they will do that specifically if there is endotoxin in that water, which that is very likely, and glucan together, because they do have a synergistic relationship. We then have the factor C coming, reacting with endotoxin. We have factor G reacting with beta-glucan, and they all sort of collide, that's the wrong word for this, but they exponentially increase the signal that we're measuring. So we see that higher signal specifically if both analytes are present, and it's seen in spike recoveries quite a bit. So you may have a situation where the unspiked sample does not have any response, but in the spike, where we add endotoxin and then the sample brings in glucan, it will cause enhancement. So in water, I think this question was about just any water. We're likely to have quite a bit both of endotoxin and glucan, and the glucan is definitely very highly likely to elevate the signal that would be measured. Yeah, I've seen that myself many times.
No, I think you're right, but I think a good place to look, and if we look back at that Kikuchi paper that I talked about, if you look at endospecies, and Veronica, help me out here because it's a Seigogaku product. My understanding, reading the literature, is that the endospecies is a TAL product from which the glucan pathway has been removed. It's been fractionated out. And so what remains of the endospecies is a TAL reagent. So if you look at the Kikuchi data and you say, okay, so here's a sort of an analogous, a regular amoebocyte reagent with no factor G pathway, how does that compare to the recombinant factor G that also has new glucan pathway? I think you'll find that they're not always the same. So it's not clear at this point, and this is why I think we need a whole lot more data. It's not clear at this point that just removing that glucan pathway is going to make everything compatible or everything sort of go the same way, or detect the same number of autochthonous, same activity of autochthonous endotoxins. So that's why I like on that Kikuchi paper, that's why I like to compare things back to endospecies, because that really is and Veronica, like I say, this is your company's product, but my understanding is that it really is a TAL lysate from which the factor G pathway has been fractionated out. So it's, I think, a really good comparator.
Yeah,
good.
That's true.
Thanks a lot for that, both of you. Our next question to Veronica. You mentioned that the recombinant reagent showed exceptional linearity. Wouldn't the linearity depend on the analyst creating the standard curve and is susceptible to human error?
Oh, yeah, absolutely. Yeah. That is a huge factor. So that's why I was mentioning the fact that there's many factors that go into the linearity. And there is the chapter in 1085 actually goes into great detail on that, and also on how we can manage that, how we can monitor the linearity, how we can control it. But if you have a truly side-by-side comparison, when you have, a well set up test, well-controlled assay, you have trained technicians, so everything is in the state of control. And the only variable in there is the reagent. That's where you can truly speak about the differences in the linearity between those reagents. So that was the example here. Yeah. And
pardon me, go ahead, I'm sorry.
I was just going to say that you can have an exceptional linearity, but you can also report complete bogus data if there is, for example, an dilution error in your standard curve. Let's say you diluted them 10 times more. The linearity is still going to be great if the dilution error carried down the dilution series. So the linearity is really just the tip of the iceberg that you should be looking at when you're looking at the standard curve.
Exactly. The y-intercept is really an important thing to be paying attention to, because very small changes in the y-intercept can result in pretty significant changes in accuracy. So in addition to linearity, you're absolutely right, Veronica, and you can have a perfectly linear standard curve, but a perfectly invalid test result because the y-intercept is off, largely because of a dilution error.
Thanks a lot, both of you. Our next question is for Karen. In the FDA inspection guide for BET, they mentioned that the turbiddimetric and chromogenic methods cannot be used with certain turbid or colored products. if the product could not be diluted and is still within its NVD, are these tests still appropriate? If not, are there other tests that you would recommend?
So if you have a test that the test material is cloudy, like I remember testing procaine penicillin G, and procaine penicillin G looks like milk. And so that one truly was not so good for turbidimetric because there was all kinds of stuff scattering the light. So that one was best on chromogenic, but we need to remember that chromogenic is also susceptible to turbidity. So even though we measure color in the chromogenic test, if you have a highly turbid sample, you can screw that up too. So what you need to do is dilute out as far as you can to get rid of any of that interference, and then choose the test that works best for you. Otherwise, you might have to go back to the gel cloud test, where the color doesn't matter.
Is there anything Veronica would like to add to that answer?
Yeah, like Karen said, the optical interference is not going to play a role with the gel cloud test, so that is the easiest way of just switching out the method if there is no way to get past that turbidity. And it does happen a lot of the times. There are also situations where when the sample is under test, it may start coming out of solution. It may actually even produce precipitate, which will cause optical interference as well. So in those situations, going with the gel cloud method is the easiest because you don't have to worry about any of that. But that being said, if we do want to, in these situations, stick with the kinetic methods, then a sample treatment would have to be implemented. So it would have to be worked out to actually set up the conditions. But then the sample treatment has to be validated as a sample treatment. So it requires a little bit more work there.
Thanks a lot. Our next question, I would ask Karen for this one. Can we make sample dilutions with NaCl during routine testing for BET?
I wouldn't. I wouldn't make sample dilutions with them. I think if you need to adjust the pH once, you might want to do that. I actually, because I'm a lousy chemist, and so I don't know exactly how much HCl would go in order to neutralize a problem. So I would use a tris buffer to do my dilutions in rather than worrying about doing dilutions in HCl. Remember, endotoxins can exist in low normality HCl. So I wouldn't do dilutions in HCl. You might want to use a more concentrated HCl in order to validate the test in the end if your pH walks out of its appropriate range. But since I'm not a good chemist, what I would do, well, I'll tell you about my stupidity. I would say, okay, I put a little bit of hydrochloric acid in and go, oh, geez, I went over. And then I'd start to drip in some sodium hydroxide and I'd say, oh, geez, I went over. And then I'd start going back and forth. The easiest thing to do is to use a buffer in order to make those dilutions.
Great, thanks a lot. Anything Veronica would like to add to that answer?
I agree with that. I think just preparing dilutions into water is the best way of doing it, and because the standard is also prepared in water, so you really want to set up the same conditions.
Yeah.
So that is usually the rule of thumb.
Great. Okay then. Our next question for Veronica. Have you seen any underestimation with, I might say this wrong, autochthonous endotoxin with your fully recombinant reagent?
What I did see is that not underestimation, but I saw the results within the 50 to 200% of the error of the test. So and this would be specifically in water samples that contain the indigenous endotoxin, it would be within the 50 to 200%. Yeah. But if anything, the tendency is generally that it would be maybe at 90 or 80% of whatever was being measured in the LAL test. And this is from results and from studies that are well controlled, where you basically run both at the same time on exactly the same plate. So the variability of the test is quite limited there.
Anything Karen would like to add to that answer?
No, I think Veronica hit it, and I think what it's really important when you do these comparability studies is that you do them as closely as possible, best on the same plate, because what you want to do is minimize all of the variables except for the reagent. And so what you want to be able to do is make sure you run the test within a certain number of hours of each other, because you don't want the sample to remain in storage for 3 days before you get to the next test. So you want to run them all together. You want to run them all against, I would think, run them all against the RSC in terms of the standard curve calibrator. And the reason for that is if you're comparing one reagent, Veronica's reagent to somebody else's, now you've got an RSC CSE calibration problem. And so if you just get rid of the CSE and calibrate everything, use RSC as the standard for the standard curve, then you get rid of all of the variability associated with that. So as you write the protocols for these studies, I would think carefully about where are the variables and how can I minimize each of the variables such that the only thing that I see in the end that might be a difference between these two methods is due to the reagent.
Thanks a lot for that, Karen. Anything Veronica would like to add to that answer?
That was great. Thank you.
Okay, then for our final question today then, firstly, there's a comment on here, wonderful presentations, both of you.
Thank you.
For the question.
Thank you very much. Veronica, you were great.
Which technique of bacterial endotoxin testing would be most ideal for testing biologics? Karen, would you like to go with this question?
Anyone that works.
Yeah.
I hate to say that. I mean, I think, and I don't have a laboratory anymore, and so I don't use this stuff. My sense is a lot of people are using chromogenic for biologics. Veronica, maybe you can help a little more. My sense is that most people are using chromogenic for biologics. But Veronica, you might want to comment a little more.
I think that is true. I have a feeling that it's probably because that is already the in-house method. So if they have for example, in process water monitoring, and they're using chromogenic method for that, because that's typical. Chromogenic methods do have the biggest presence, I would say, globally among the methods in the world, because they produce color, and we all can relate to that. It kind of reminds us of the ELISA methods, right? So I think that's probably one of the reasons. So if it doesn't work with the chromogenic method, or there is a problem with the method development on that, then I would say the turbidimetric method is actually a safe bet, because of it actually does suffer from interference less a lot of the times. So it's the second choice. And that is generally in areas in countries where the kinetic assays are the preference. And then there is parts of the world where JoQuat is still the king, and that would be used for any assay and for any type of sample. So yeah, it could be. The important part is running the suitability testing. That really comes down to, and doing it properly. And so whatever is the output of that is then the right method to use for the testing.
Okay then, thank you audience members for your questions. And before we finish the webinar, I'd just like to ask our presenters if they have any closing remarks. Karen, perhaps you might like to go first.
Well, I want to thank everybody for dialing into the seminar. This was the webinar. This was a lot of fun. I enjoyed doing it, Marcus. Thanks to you and to Biopharma Asia. Always, as my friend Veronica, we've done this a number of times, and I thank you. And it's always a fun time to do these things with you. Thank you very much.
Thank you, everyone, for being here. And we're very excited to always share the knowledge and the news in the field. And we continue to look forward to more webinars in the future.
And I would like to take this opportunity to thank our presenters. So once again, that was Karen Zink-McCullough, owner, principal consultant at MMI Associates, and Veronica Wills, manager, technical services at Associates of Cape Cod. Thank you, both of you, for sharing your knowledge and your expertise with us. And lastly, I would like to remind our audience that you can watch this webinar and other webinars on demand by visiting our website, which is biopharma-asia.com/on-demand-webinars. And if you're watching this on demand and you have more questions, please feel free to send them over to me. And my email address is marcus.ossay@biopharma-asia.com, and we'll get those answers over to you. My email address should be displaying just below your webinar screen right now as well. So with that, thank you everyone, thank you audience members, and goodbye. Thank you Karen and Veronica, goodbye, and have a great day.
Thank you, Marcus. Everybody have a good day, evening, morning.
Goodbye.